Hello, welcome back to Bioclass Bytes. In this video, we are going to talk about cell cycle, specifically interphase and its stages. Don't forget to subscribe to my channel and like and share this video. This lesson is part of Grade 8 Biology, Unit 7, the cell cycle. So these are the lesson titles under this unit: Interphase and its stages, Mitosis and its stages, Gametogenesis, Oogenesis and Spermatogenesis, and Meiosis and its stages. For this video, we are going to focus on interphase and its stages. In our previous lessons, we have established that one of the major characteristics of life is cellular organization. It means that all living things, all known living things are made up of cells. In our previous lessons also, we have established that living things can either reproduce asexually or sexually. So for unicellular bacterial species, binary fission, or their mode of cell division, is also their way of reproducing, while for multicellular complex organisms such as plants and animals, the union of the egg cell and sperm cell actually is the starting point for the creation of a new organism. So how do cells reproduce? Okay, so we know that our body, the human body, is actually made up of hundreds and hundreds of trillions of cells. And if we actually trace it back, we all derive from a single cell, and that's the fertilized egg cell. So in this image, we see the comparison of the size of the sperm cell and in comparison to the size of the egg cell. So the several human sperm cells here are seen attempting to fertilize a human egg cell. So right after the union, or right after the moment of fertilization, the zygote, now called zygote, will undergo many millions of successful cell divisions in order to grow and reach the present state of your body right now. So here again is this statement. It keeps on appearing because I want to emphasize this concept. All living things grow and reproduce. In this image, we see here even cells, even something as small as cells are also capable of reproduction. Here they are seen undergoing cell division, and life without reproduction is not possible because no matter how long the lifespan of an organism is, it will eventually die. So it must have the ability to reproduce. So all living things produce offspring like themselves and pass on to them the hereditary information that makes them as they are. So I would like to share with you here my picture of my family. So if you notice, living things such as humans have the ability to pass on their genetic information to their offspring. So you see here my father and my mother and my brother. If you notice, some of the physical traits between my brother and I are actually the same physical traits seen in our parents, just in different combination. So organisms are actually products of two copies of DNA. So half, for example, in the case of my brother, half of his genetic material came from my mother, and the other half came from our father. So the sperm cell and the egg cell from these two parent organisms created this whole new organism. And even if my brother and I came from the same parents, we have genetic variation between us. We have differences between us, and imagine more in a larger group of people. So because of sexual reproduction and because of genetic diversity, there's differences, there's variation among groups of people within a species, and that's very important because it allows the species variety, and it allows the species to survive because of those variety, to survive the changing environment because of the varieties of its population. So in this unit, we will begin our consideration of heredity. So that's the concept of passing off of traits, with the examination of how cells reproduce themselves. So if you actually, when you go to grade 11, okay, we have this lesson on geologic timescale, wherein we can actually trace back. Okay, we can actually trace back the history of life, or the origin of life on our planet 4.5 billion years ago, to that single organism, okay, or a unicellular organism that underwent successful cell division. And as those billions of years go by, it eventually diverged, evolved, adapted, and become different forms of species that we see today. So the ways in which cell reproduction is achieved and their biological consequences have changed significantly during the evolution of life on our planet. And the ability of cells to reproduce is actually one of the crucial points, one of the crucial reasons why life was actually possible. Because again, we can all trace our ancestry to that last universal common ancestor, which was actually a microscopic, as a unicellular microscopic organism um, billions of years in the past. So in order for us to visualize how cells reproduce, we will use this model called the cell cycle. So this is the cell cycle. So we define the cell cycle as the series of events that take place in a cell leading to division and duplication. So as you can see here, these are the major parts of the cell cycle. So from all these blue regions, all these blue segment, that's interphase, okay, which we will discuss thoroughly. Then M phase, this is the actual mitotic phase or the actual cell division phase. So wherein the cell divides into two. Within the interphase, it's further subdivided into stages. We have here gap 1, S phase, and then gap 2 or G2. Then outside the interphase, you also have a possible gap 0 or G0 resting phase. Okay, so the cell cycle is required for cell growth and cell division into two daughter cells. So this is the product into two daughter cells. So pop quiz, let's try. I hope you were able to research before watching this video, but if not, let's try and find out. So what are the events or key events that happen in every stage of interphase? Okay, so interphase, this is the longest period of the cell cycle. So during gap 1 phase or G1 phase, this is the time wherein the cell grows in size. Okay, so this actually is done by a freshly divided cell. So in order for it to divide in the future, it must first grow in size. So it must first grow in size because it just came from cell division. So during gap 1, the cell will grow in size. Then it will enter S phase. S there means synthesis, okay, or creation, synthesis. So during S phase, the DNA is synthesized or the DNA is replicated. Once it's done, the cell will now enter gap 2 or G2 phase. So here, this is nearing mitotic phase or division phase. The cell prepares everything it will need for mitosis. Okay, so growth, duplication of DNA, and then preparation for mitosis. And then on M phase or actual cell division, so M phase or mitotic phase, the cell divides into two new cells. So these are the breakdown of the stages of the cell cycle. So in order to visualize the different stages of the cell cycle, I recommend that you watch this video entitled Animation How the Cell Cycle Works. I'll provide the link in the description below. So the cell cycle varies between the type of cells, okay? So for prokaryotes, the cell cycle occurs via the process called binary fission. We have established this in our lesson on asexual reproduction. While for eukaryotes, the cell cycle is divided into three periods. The interphase, during which the cell grows, accumulates and nutrients needed for mitosis, and duplicates its DNA. And then the actual cell division, during which the cell splits itself into two distinct cells, often called daughter cells. So in comparison, the cell cycle of prokaryotes are simpler, okay, compared to the cell cycle of eukaryotes, okay? So for them, it's just one process, binary fission, and done, there are now two cells. While for eukaryotes, the cell must undergo interphase and all its substages before it performs the actual cell division. So I recommend that you visit this lesson from Khan Academy, The Phases of the Cell Cycle. I'll provide the link in the description below. So for prokaryotes, binary fission, okay, fission, separation, division, binary into two, okay, or also known as prokaryotic fission or separation or division, is a form of asexual reproduction in which the cell just literally divides into two. Okay, so this is used by all prokaryotic organisms, all organisms with no um, established nucleus, okay, nuclear region. They just have a primitive nuclear region. So here, as you can see, the cell just elongates and then it divides into two. So I recommend that you've watched this video, Binary Fission HD Animation, for a more detailed explanation. I'll provide the link in the description below. And this video, okay, of E. coli growth and binary fission, and you will see how fast they reproduce, because asexual reproduction is actually more efficient than sexual reproduction. So you will see in this video how fast they reproduce. Again, I'll provide the link in the description below. For eukaryotes, I recommend that you visit this video, watch this video entitled Eukaryotic Cell Cycle Biology Genetics, a more thorough explanation of the different stages of the cell cycle for eukaryotes. I'll provide the link in the description below. Now, this one is a fun interactive website from cellsalive.com. So here, this is actually an interactive a presentation wherein you can fast forward the cell division. You can click on the stages of the cell cycle and then find some information. So very fun to visit. Okay, again, I'll provide the link in the description below. So what are the stages of the cell cycle? As what I've mentioned in this video, we will be focusing on the stages of interphase. Okay, so interphase covers gap 1, S phase, and gap 2. Okay, so as you can see, this is actually quite the longest period of cell cycle, and it occupies almost almost 90% of the entire life cycle of a cell. Um, um, so actually, there used to be a misconception that the cell is actually inactive during interphase, during this phase. But in reality, as our technology to observe cells become better over the years, we see actually that there are a lot of activities going on inside the cells during interphase. So the first one is gap 1 or G1 phase. So here, cellular contents, excluding or except the chromosomes, are duplicated. So during gap 1, everything are duplicated, and this is the time that the cell increase in size. If you're wondering why does the cell have to increase in size, do not forget that right before gap 1 was cell division. So we can assume that the cells are actually smaller during this phase in terms of size when compared to an original parent cell. So since they're small, they have to grow first, they have to enlarge first before they undergo another division. They cannot keep on dividing without growing, or else after several generations the cell will just disappear. So since they're just small here, they have to grow and develop before they divide again. Okay, so that they can return to their original size before they divide into two smaller cells. So that happens during gap 1. During S phase, each of the 46 chromosomes is duplicated by the cell. Okay, so this is anticipation of future cell division. So the 46 chromosomes are duplicated, are synthesized during S phase, and then by gap 2, the cell double checks everything it will need for the actual cell division, for mitosis and cytokinesis. So S phase, the DNA, the chromosomes are duplicated. At gap 2, everything is prepared for the cell division. And on mitosis or M phase, this is the actual cell division stage, or the different substages will be discussed further. So this is where the cell will actually divide, but mostly focusing on the chromosomes and the nucleus. While the division of the entire cell, of the cell membrane and then the cytoplasm, will happen afterwards. Um, in cytokinesis. So these are the stages of the cell cycle for eukaryotic cells. Interphase, which covers gap 1, S phase, and gap 2. Then actual cell division or M phase called mitosis, or the division of the nucleus. And then cytokinesis, the division of the entire cell, cytoplasm and cell membrane. Now we also have another stage here called gap 0 or G0, wherein the cell completely leaves the entire cell cycle, or it stops the processes of the rest of the cell cycle. So they do not continue with the cell division anymore. And examples of such cells can be brain cells, heart cells, and bone cells. So for those kinds of specific cells, the cell cycle is arrested. So these are a further description of the stages of interphase. So again, for gap 1, so the cell just came from cell division. The cell increases in size. So in gap 1, the G1 checkpoint control mechanism ensures that everything is ready for DNA synthesis. Now, gap 2, this could happen an arresting phase where the cell has left the cycle and has stopped dividing. So if the cell decides to continue, then it will undergo S phase or synthesis phase. DNA replication occurs this phase. So the 46 chromosomes are duplicated. Then gap 2, during gap 2, the cell will continue to grow, and the gap 2 checkpoint ensures that everything is ready to enter the M phase or the mitosis phase for cell division. And M phase or mitosis phase, the cell growth stops and the cellular energy is focused on the orderly division into 2 daughter cells. So I recommend that you visit this website from Virtual Genetics Education Center for more information and more details about the cell cycle. So I'll provide the link in the description below. Now, these are important concepts, okay? So for simple unicellular organisms such as the amoeba being shown here, amoeba proteus, one cell division is actually equivalent to reproduction. So once they undergo cell division, they have reproduced already and they are now counted as 2 organisms. Okay? While for larger organisms such as plants and animals and humans, mitotic cell division is important because it can create offspring from multicellular organisms and also allows plants to grow, although allows the organism to grow. Okay? Well, for some animals such as plants, they use mitotic cell division to grow from cuttings or to grow from vegetative propagation. So for larger organisms, mitosis is mostly used for growth, while for microscopic unicellular organisms, they use cell division for reproduction. So now let's look into the actual stages of cell division. So the previous ones are mitosis, are stages of interphase. Now let's look at the stages of cell division. So we define cell division as the process by which one parent divides into two or more daughter cells. So we have established that for prokaryotes, that their cell division is called binary fission. We've seen the GIF. Hopefully by this time you've seen the recommended videos. For eukaryotes, there are 2 types of cell division, so mitosis and meiosis. Mitosis are usually performed by body cells, and this leaves the daughter cells capable of dividing again. So after mitosis, the cell will undergo interphase and its substages, and then it can divide again in the future. And these are performed by body cells such as your skin cells, your muscle cells, your brain cells, your heart cells, and all internal organs. Okay, your smooth muscle cells. So they perform mitosis, and they can divide again in the future. However, for meiosis, okay, this is also done by eukaryotes. Meiosis, the cell is permanently transformed into a gamete or into a sex cell. So for eukaryotes, the products of meiosis are either egg cells or sperm cells. However, those sperm cells and egg cells cannot divide anymore. They cannot divide again until fertilization. They are just produced by the body with the hope of reproduction. So if they are not used for reproduction, if they were discarded, or if they were not utilized, and then they will just be disposed. So before we continue with the different stages of mitosis, these are some of the terms we have to clarify and understand because we will keep on repeating them throughout the lessons. Okay, so first, chromosome. So it is an organized structure of DNA and protein that is found in cells. Centromere is a region of DNA typically found near the middle of a chromosome, and it actually attaches sister chromatids together. Spindle fiber is the structure that separates the chromosomes into the daughter cells. Centrioles are cylindrical structures that organize the assembly of microtubules during cell division. Mitosis, again, is the division of body cells into two diploid daughter cells, while meiosis is the division of sex cells into four haploid daughter cells. So a chromosome, again, is an organized structure of DNA and protein found within cells. Um, so so I hope you still remember this is the cell, and then cytoplasm, and then the nucleus. So within the nucleus, you can actually see this visible condensed structure that looks like letter X here. So those are chromosomes, okay? So this one is a chromosome. Actually, if you look closely, chromosomes are condensed, okay? Condensed or compacted strands of DNA, okay? So if you actually unravel the chromosome, you will see that it's actually made up of threads of DNA. And this thread of DNA or strands of DNA actually coil around proteins called histones, okay? So the DNA strand is coiled around histones, and they are found compacted in the chromosome, and chromosomes are found inside the nucleus. So again, as a chromosome is a single strand or single piece of coiled DNA containing many genes, regulatory elements, and other nucleotide sequences. So we have here a video. What is a chromosome? So I will provide the link in the description below. So please watch this, very, very informative. So here, the video defines chromosome as an entire chain of DNA along with a group of stabilizing proteins. So these are spectral karyotyping images of the human chromosomes. So they're not usually this colorful, no? They were just um, colored this way to see them clearly. So this is how our chromosomes look like. And if we actually arrange them, we can create a karyotype. So a karyotype is the number and appearance of chromosomes in the nucleus of a eukaryotic cell. So this is the karyotype of a human male. So we have a total of 23 pairs of chromosomes. So that's a total of 46. So 23 pairs, so 1 to 22, then the 23rd pair or the 23rd chromosome, that's the sex chromosome. So if you multiply that into 2, so we have 46 chromosomes all in all. And as you can see, we know that this is a chromosome, this is a male karyotype because the 23rd chromosome or sex chromosome is XY. So it tells us the 23rd chromosome, it tells us that it is a male. In comparison, this is the karyotype of a human female. So same number of chromosome pairs, 1 to 23. Okay, but as you can see, the 23rd chromosome are both X. So XX or 2X. Okay, it tells us that it is actually a human female chromosome. Okay, so that's how our chromosomes look like. Centromere, on the other hand, is the region typically at the center of the DNA, at center of the chromosome. So it's a coil of DNA. Okay, and this is where two identical sister chromatids come together or attach together. So again, no, let's study the structure of a chromosome. So this one part, okay, or one arm, that's one chromatid. So if there's two of them together, two of them identical together, this is one chromatid, this is another chromatid, we call them sister chromatids. What attaches sister chromatids together is centromere. number 2 at the middle. Okay, so the part of the chromatids above the centromere is the short arm of the chromatid, and then the long part below, that's the long arm of the chromatid. Okay, so as you can see here, so for example, if the centromere is found here, this part is the short arm, then this is the long arm. Okay, so the centromere attaches two sister chromatids together. Spindle fiber, as you can see here, it's the pinkish colored here structure. They are the ones that pulls the chromosomes apart during cell division. So they separate the daughter cells during cell division. So it is part of the cytoskeleton, okay, as you can see here. And again, they pull the chromatids apart. So the chromosomes here, the chromatids here are the violet colored parts. Okay, so it's quite beautiful to look at. So the sister spindle fibers, they pull the chromosomes apart. Then the darkly stained colored violets here, structures, those are the chromosomes. So centrioles are cylindrical structures found in animal cells, and they help organize the assembly of microtubules during cell division. So usually you can find centrioles at the edge of at the opposite edges or opposite sides of the cell undergoing cell division. So centrioles, this is where the spindle fibers are connected, and this is where the chromatids will be pulled, or the chromosomes will be pulled towards mitosis, okay, towards the end of mitosis. So they are found at the opposite end of the cell during cell division. So they look like small tubes like this. Okay, that ends our first video on this series on cell cycle. I hope you learned something new. Don't forget to subscribe to my channel and like and share this video. Until next time, goodbye.